Welcome to the Gladden Longevity Podcast with Dr. Jeffrey Gladden, where our passion is helping you become an age hacker. On this show, we want to help you optimize your life energy, longevity, health, and human performance with impactful and actionable information by answering 4 questions. How good can we be? How do we make 100 the new 30? How do we live well beyond 120? And how do we live young for a lifetime? I'm Steve Reiter, and Dr. Gladden, we just wrapped up with Liz Parrish with BioViva Science and talking about gene editing or gene therapy, really.
Gene therapy. Yeah, not gene editing, but gene therapy. I think this is really a fascinating area for longevity, right? It's really how do you get the genome to express itself in a youthful way? And she's going to walk us through thoughts on that. She's going to walk us through what she's done to actually insert genes into her own cells. And we'll hear all about this and kind of some of the net effects. It's really a super exciting show. I found this to be really, really interesting conversation. So
our listeners are familiar with telomeres and the length of your telomeres and the importance of that. The increase that she saw was nothing more than radical, as well as what you've seen in your telomeres.
Yeah. We're coming at things from different angles and getting similar results, but there are some advantages to what she's doing that are really, really intriguing. I want to explore more with her. So fun stuff. Yeah.
And for our listeners that this may be your first Gladden Longevity Podcast episode, we want to say thanks for tuning in and thank you for listening. And be sure to click that subscribe button in your podcast app, because that is the way that you will be able to stay on top of these things. And if there is something in one of our episodes that really speaks to you, please consider sharing it with a friend.
Yeah, and really helps us a lot if you'll leave us a review. We get lots of comments about how much people enjoy the podcast. And if you leave us a review like that, it helps us to really expand the listenership. It moves us up in certain ratings and rankings that I don't pay a lot of attention to, but we know that we want to try to reach more people. And so we really appreciate your support and supporting us in that way. So thank you for that. Welcome, everybody, to this edition of the Gladden Longevity Podcast. I'm your host, Dr. Jeffrey Gladden, and I'm here with my compatriot, sidekick, and co-host, Steve Reiter. Steve, how's it going?
It's going well. Had a great weekend. Got a nice hard 4-mile hike in with my dog on Sunday and got some outside time, enjoying when we're recording this, it's October. So enjoying the nice October weather here in Colorado Springs. How about you? You're back in Dallas now, right?
I am. Mukti and I came back on Sunday, but we had a wonderful weekend in Puerto Rico. Really, really special, fun time. So it was great. Yeah. Outside, doing some really cool things. We started playing games down there. I got a ping pong table, cornhole, some kadima. I think I've told the audience, I've started doing Justin Franson's, who was on the podcast, wrote the book Athleticism. I started doing some of his infinity walking, his figure 8 sort of walking with your eyes closed, with your head up, look, kind of like head at different angles, walking frontwards and backwards.
What does that do?
It's fascinating how it activates your entire nervous system, right?
Really?
Yeah. Health is really about training the nervous system in the end of the day in terms of performance and everything else. So he was talking about making yourself ambidextrous by doing these kinds of things. So I've got into it, and it's almost like you're in the middle of ecstatic dance because you're kind of going faster and you're fluid. And I do it to wake up my nervous system before I mountain bike, and I'm just like so fluid with the bike. It's like so cool.
Really?
Yeah. And so I started playing ping pong with Mukti, and I thought, what, I'm going to play with my left hand. So I started playing with my left hand, and it's like, I'm not actually bad with my left hand. So I was going back and forth between hands. It was very, very cool. So I love this kind of stuff. Yeah. So today we have somebody with us that plays in a big space in the whole world of genetic engineering for longevity, quite honestly. It's Liz Parrish. She's the CEO and founder of BioViva. And I bumped into Liz at RadFest when we were both there about a month ago. We didn't get to chat at that point in time, but she's agreed to come on the podcast. And Liz, welcome to the show.
Thanks for having me. I'm so interested in this figure 8, head up. What do you mean head up? Like chin up, like eyes closed?
Yeah, like eyes closed, looking up with your head at different angles, and you're walking in a figure 8, right? So your body is doing, right? And it's so cool. You should try it. I mean, you might want to do it in an open space where you're not going to bump into anything.
Yeah, I remember reading that, like, when you swim, if you swim with your eyes closed, you can't swim in a straight line or something like that. And so I guess with your feet on the ground, it's different. You're able to achieve the figure eight.
You can actually do the figure eight. And it may not be a perfect figure eight, but it doesn't matter. The fact is your nervous system is starting to learn where you are in space and how to move. And that's the whole joy of movement, quite honestly, right? We do everything kind of like this, and it's too restrictive. The nervous system likes to be liberated from that. So what's new at BioViva? We talked about 4 or 5 years ago, I think, but you've been on the move with a lot of different things. And quite honestly, you look better today than you did 5 years ago. So whatever you're doing is working. But what have you been up to?
Thank you. Well, I've done a couple of gene therapies, but as far as the company, so we are pushing towards a money raise now so we can get into a phase 1B for dementia.
Okay.
We're using the data that we assessed that Integrative Health Systems procured from 5 patients who underwent a dual gene therapy. And then we're also working on...
So let me interrupt you for a second. So just so the audience understands, so, there is a genetic component to dementia. There's also a lifestyle component to dementia. There's multiple components to dementia. But there is definitely a genetic predisposition for dementia. So when you talk about people that have had gene therapy, what genes actually were you going after? What was their genetic profile and what genes were you going after?
Right. So with dementia, it's actually really interesting because some of the genes that predispose people to be in high risk for dementia are also associated with longevity. And so there's a lot of strange things going on with dementia. But when we're looking at patients who already have dementia, there's little that we can do right now about the genes that maybe predispose them and then cause the symptoms and have landed them in the midst of the disease. Mm-hmm. So what we look for are genes that might be able to recapture some of their function. And of course, since we look at treating aging cells, think about it as like targeting the hallmarks of aging. We don't entirely do that, but those are great targets for therapeutic intervention. We use genes that should make the cells behave more youthfully and therefore hopefully reverse the condition of the diseases themselves.
Okay. So would these people have like an ApoE44 or ApoC1 or these kinds of genes? It is interesting, just so the audience understands that when you start playing with genes, you don't really know what the full extent of their benefits are, even though you're aware of perhaps what their liabilities are. So it's kind of really interesting to think about going in and manipulating that.
Right. So we don't actually edit genes right now. What we do is we add genes to the cells that are associated with cells behaving more youthfully and combating some of the effects of Alzheimer's. So one of the genes we look at is Klotho, and people upon autopsy who had Alzheimer's upon autopsy but didn't suffer the cognitive decline are alpha Klotho upregulators. So this would be a protective gene and increases the cognition in animal studies, monkeys and mice, and we think humans, because that's what we saw in the study. Another gene target is telomerase reverse transcriptase. Mm-hmm. It's associated with reducing tau tangles and creating healthier cells that function better. And it also protects mitochondrial health. And then a third candidate is a gene called PGC1 alpha that basically is associated with mitochondrial biogenesis, therefore upregulating the energy in the cells. And that gene is associated with better cognitive outcome for Parkinson's.
Nice. What's interesting is we're actually doing something similar to what you're doing in the sense of we're doing transcriptomic and proteomic testing inside of our IRB-approved trial called LifeRaft using a test that we've developed called the LifeRaft Index, where we're now looking at 8 of the drivers of aging, we'll call it, including oncogenic potential and some things that aren't traditionally part of the hallmarks. And we're going to expand that by about another 10 this coming year. But then we're actually working with a company to create HLA-matched custom peptides to go in and modulate genetic expression to both upregulate and downregulate, again, with the idea of trying to get the genes to to basically be expressed in a more youthful pattern.
Mm-hmm.
And there's also a bit of durability to the technology. It's currently used in the cancer space to basically shut down genes. And here we're not trying to shut down genes. We're basically trying to modulate them, right? But in doing so, the daughter cells, from what we understand from the research, is that the daughter cells of these cells that are exposed to these peptides actually carry on that same modified genetic expression.
Mm-hmm.
So now we're actually getting a more sustained, we think, we're getting a more sustained genetic reprogramming without having to do CRISPR, where you splice out a gene or add a gene per se. Although, we're also looking at how do you upregulate, things like TERT and things like clotho and other things like that. So we're kind of coming at a similar thing from different sides of the fence, so to speak. So it might be fun to put our heads together on this. We can
absolutely. Yeah, we really love the gene therapy approach because it does upregulate the gene for the life of the cell without integrating. So
right.
What we don't want to do is we don't want to mess with the natural genome to the human cell. We're just basically, adding this little episomal piece of DNA in there to upregulate just the protein that we want and then see what happens when we do that. Of course, we're actually professionals at combinatorial gene therapy. So, we would rarely use a single gene.
Right. And that's a good point, too, just for the audience to understand. Everything in biology is so interconnected in such an elaborate fashion with so many feedback loops and things that amplify and de-amplify a particular signaling. It's incredibly complex, particularly when you add the environment into it. So I'm curious, how did you choose these genes and what kind of results are you seeing with what you've done?
So how we chose them is we did a meta-analysis of all the gene work that had been done. And we assessed it, and then we look at mechanism of action to the best of our understanding, and we try to work with genes that would bolster one another, that might actually end up with a higher net effect. So when we started the company in 2015, we thought that telomerase reverse transcriptase alone may reverse all of aging. And of course, it was everyone's hope that it would. It might have been very convenient.
Yeah.
And to this day, we have never been able to deliver the gene to every cell in the body, but it could be assumed that we need supporting roles and that we need to hit more targets, the hallmarks of aging, which for a single gene delivery, telomerase hits the most of them, but our other gene suspects are, they have a beneficial effect on the overall health of the body, either in a geroprotective mode or in a direct target of the hallmarks of aging. And so we have about 12 gene candidates now in the company.
Nice. So 12 gene candidates. And I'd be curious to see the list. Do you want to try to rattle them off, or shall we put them in the show notes?
No, I don't want to try rattle them off. So what we do at BioViva, so we started in the AAV delivery space, which was a well-known and well-understood gene therapy.
AAV, just so the audience knows what that is, we're talking about a viral vector. Is that what we're talking about?
It's adeno-associated virus, and it's the vector behind all the approved gene therapies today that are on the market. There's 12 in the U.S. They just approved 3 this year, which is the fastest rate they've ever moved at.
So essentially you put a gene in a virus and you let the virus infect the cell with that gene. That's really what's happening here.
Yeah, they're attenuated, so they cannot get you sick. And I think that's the most important thing for a lay person to understand. These are not viruses really anymore. That's why we call them vectors. What they have left, we've sort of obliterated all of their parts except for their ability to connect to a cell, a docking mechanism, and insert genetic material. So we've taken what nature does naturally every year over and over to us and other species on the planet, and we have basically re-engineered it to deliver genes that make you healthy.
Yeah, nice. Yeah, so this is very cool. So I'm curious, how many people have done this, and where are you doing it?
So as far as my company, we can't treat patients. We're a U.S. company, so we have to do clinical trials. So we're in a long queue of waiting to be approved for phase 1.
Okay.
But there are companies that do things like this. There's one in particular called Integrative Health Systems.
Mm-hmm.
And I think they don't treat patients either, but they connect patients with medical doctors that can help you get access if you qualify for a treatment. And we assess that data. And those are called investigator-led studies. So they are legal studies. They're done as a study would be done, except for without the enormous costs of a clinical trial. And then when we get the data, we find out where these drugs work, how they work, and where they don't work.
Cool. And you yourself, I think, mentioned that you've had some gene therapy done. Do you want to tell us a little bit about, well, as much as you're willing to reveal in terms of what you thought your genetic hurdles might be and why you chose to do the gene therapy that you did and what kind of results you've experienced and whether or not you required more than one procedure? Just kind of walk us through that a little bit if you don't mind.
Yeah. So how I ended up here was not trying to live forever. It really wasn't thought in my mind at the moment. My son was diagnosed with type 1 diabetes in 2013.
Okay.
And I had spent 2 years piecing together what was happening in the stem cell industry for a project that was on the education of the use of stem cells and how to get the technology funded into patients that needed it.
Mm-hmm.
So I had learned about all this fabulous regenerative medicine. And when my son was in the hospital, they told me that was all experimental medicine and it wasn't used in children as if it was a dirty word.
Right.
And, but in the meantime, when you're there, you hear coding and there are actually kids dying and we don't use what's called experimental medicine. But, it came to my mind that it seems like all medicine is an experiment and a whole bunch of it is failing us and our children.
Yeah.
So I got on one plane after another looking for cures for kids. And I ran into a conference that was going to be speaking about aging. Professor George Church from Harvard was going to be there. He's a professor of genetics. And I thought, well, here is a place to go find out what's happening. It was about aging. And it took me about 3 days with jet lag to fit that into my head of how this could actually expedite curative medicine to children much faster by targeting the biggest medical unmet need, and how 80-year-olds are just 8 times 10. They just feel like kids themselves. We don't have enough time to grow up and make the right decisions, and that maybe what the world needed is life, health, extension. Mm-hmm. And George Church, maybe 2 years later, became a co-founder of BioViva because we shared that desired mission to cure disease through combinatorial therapies that would help everyone live longer and healthier.
Mm-hmm. And so what were you facing besides your son's scenario did he actually have gene therapy done or?
No, he has not. He has not elected to do any experimental therapies yet. He does have a closed loop system and a Dexcom, and he mm-hmm. has, there is one approved cell therapy in the United States that was just approved a few months ago, and he's going to ask his doctor about that in a couple of months.
Okay, nice.
He's a little bit less experimental than I am, which is,
right.
His prerogative.
Right, exactly. Yeah.
So you asked me what I did.
Yeah, I did. I was just thinking about the fact that you're experimental. I think we probably are cut from a similar cloth there. So it's kind of like, let's see.
Yeah, well, I basically walked out after 2 years of research with the 2 most promising gene therapies at the time, telomerase reverse transcriptase that had helped mice live longer and healthier in a multitude of studies. Yeah. And follistatin, which was a gene that had already been used all the way through non-human primates and in Becker's muscular dystrophy through safety and efficacy for muscle loss.
So just so the audience knows, the telomerase is going to re-lengthen or maintain length of telomeres, which would be critical. And you particularly would want that in your stem cell population, which tend to maintain telomeres depending on the type of stem cell you're talking about. And the follistatin is actually a myostatin inhibitor. Myostatin is an enzyme that breaks down muscle. So if you lift weights for a month and you add muscle, but then you don't work out for a month, the muscle doesn't stay, it goes away, right? And the reason it goes away is it's a high metabolic cost to the body to hold on to that muscle. It's got to feed it. It's got a big metabolic cost. So to be economical, the body trims away unused muscle. That's why when you stop working out, myostatin goes to work and you lose muscle mass. So follistatin comes in and blocks blocks myostatin and augments muscle growth. In fact, there are some kids that are born with mutations, I think, in follistatin that are like these 6-year-old kind of like Hulks, right? With big muscles.
They're the myostatin knockouts.
Or the myostatin knockouts, yeah. Okay.
Yeah. It also has great implications on metabolic health. So as you know, if you increase muscle mass, you increase insulin sensitivity and probably a lot of other beneficial aspects around metabolism, which was not what we were thinking at the time. We were thinking more around frailty, but then we came to the realization of the metabolic attributes. And the gene therapy is actually the only thing that can lengthen telomeres that we know of predictably. And it's what the companies use generally to make small molecule drugs. They tested against the gene therapy expression. So those were really the telomerase reverse transcriptase was a major highlight. So in 2015, to start the company, we started it with the big idea that we would use these drugs in humans and get them to the mass population as soon as possible and revolutionize healthcare. But of course, that's more difficult to do.
Right.
Us young, wet behind the ear babes thought. And but we did start with me taking the first 2 gene therapies to treat biological aging.
So what year did you do that in?
2015.
Okay. And so what was the net effect for you? What did you notice?
Well, we actually released a paper about the telomeres part of it because I redid the gene therapies in 2020 and added Clotho and PGC1 alpha.
And why the repeat? Why the repeat on the other ones?
I got an opportunity, and it was an opportunity in which somebody who was investing said, how would you like to redo some of those therapies? And what do you want to try new? And I said, yes, absolutely, I do. Because the first time I did telomerase reverse transcriptase, it was mostly injected locally in places that we thought were high cancer risk.
Okay.
So I didn't get to really actually have a full systemic dose of the gene therapy. And it was something that I really wanted to.
Okay.
Since with gene therapy, we can't target all the cells in the body, a gene like telomerase reverse transcriptase probably is best given every 5 years and sort of layered into the system. Whereas something like follistatin, PGC1 alpha, and Clotho have what's called a shared protein. They will actually excrete it into the body, and the whole body benefits even if you only treated one area.
I see. Okay. So is the immune system treated with the telomerase gene?
Well, the thing is, when you do a test like this, so in 2015, all the biological aging tests mostly didn't exist, though the one that did was telomere length in the T lymphocytes. And so that is really where we have our data. So, what can you do post to a test? You can do a lot of things, but if you don't have any pre-data, it's pretty much meaningless. So what we had was all of my historical blood work and then blood work right before, my C-reactive protein, my MRI of my legs and for muscle mass testing, and then again, the telomere length in my T lymphocytes. And that's what we follow ever since then. And that's where we see increase year by year. And then we had another big bump in 2020 when I did the gene therapies again.
This episode of the Gladden Longevity Podcast is brought to you by H2 Molecular Hydrogen in the Gladden Longevity Store.
Yeah, Steve, you've heard us talk about this numerous times on the podcast in terms of the benefits of H2 and hydrogen water. We love it. It's really the best way to balance your entire redox system. So it really protects you from free radical damage. I take it every time I get on a flight to protect me from radiation damage when I'm flying. And I use it to rejuvenate my brain in the afternoon. There's really 2 kinds of exercise. There's physical exercise, and taking H2 before or after that works great. But also there's mental exercise. And I find that the H2 will bring your brain back when you're tired in the afternoon. So I think it's like the perfect thing for longevity and for performance, quite honestly.
And I talked to you about this, that I found dropping 1 or 2 tablets in a glass of water right before bed. I've seen an overall bump in my deep sleep when I do so. And
fascinating. Yeah.
Yeah. So if you click on the store tab when you visit GladdenLongevityShop.com and use the promo code podcast10, you'll get 10% off your order. And this code isn't only good for our H2 product, but it's also good for all the supplements we carry. So podcast 10 at the GladdenLongevityShop.com.
Yeah, one of the other things that I use it for routinely is when I'm about to go on a mountain bike ride. And I will say this, that if I go on a long mountain bike ride, I will take one prior, I'll take one during, and I'll take one after. And some of the Ironmen that we've worked with have said that when they're doing an Ironman, they'll take it 3 or 4 times throughout the race. And their comment is that it's like taking out the old battery and putting in a new one. It really rejuvenates your body's ability to perform when you're doing more extended athletic activity. So think about that too. If you want to go do a longer hike or whatever else, take it with you.
So GladdenLongevityShop.com, enter podcast 10 for 10% off your order.
H2 molecular hydrogen. It's low-hanging fruit on your quest to make 100 the new 30. Okay, so let's talk about a big bump. So are you saying that you didn't have telomere lengths measured prior to the initial gene?
Yeah, we did.
You did? Okay, and what was the increase in your mean telomere 20 percentile?
I have my shortest telomeres, my average, and my mean, and I don't have them right in front of me, but I know that my shortest telomeres, that was one thing we did at RadFest. We showed the average length increase in the 20th percentile shortest telomeres in 10 patients.
Yeah.
I believe my data was one of those over some years capacity from treatment till now. And the average was 1.22 kb. Okay,
1.22. Okay, I'm going to show you something here. We can keep talking. Okay, so, that's really interesting. So let me ask you, what have you experienced in terms of how you feel or function, or have you noticed any difference that way, energetically or physically, or how you look or how you feel or what you're capable of doing, or any endurance benefits?
Yeah, I think that was the biggest impact of the therapies were performance. We saw changes in triglycerides, C-reactive protein, blood glucose, the HbA1cs to the benefit. But at the day's end, I was able to achieve all of that without modifying anything in my lifestyle. As a matter of fact, my lifestyle became much worse after doing the gene therapies because I was traveling. I went from, jogging 5 days a week to not exercising because, it was traveling around the world, speaking to ministers of health, speaking at conferences on why I thought that this was an important thing to do. And my overall health was better. I always say that like the people fighting for your longevity probably won't obtain it because we live such a stressful lifestyle, but I seem to be doing fine.
Right.
My average or mean telomere length went from 6.15, I think, to 9.33 in SpectraCell. And then in life length, it went to over 12 kb. So those two companies measure the telomeres different. And even though they have a completely different style of test, they're both consistent with each other. So that was interesting. So larger gains in the average and mean telomere length.
So I have some telomere. We have a telomere product that we've been developing also that delivers telomerase into cells. And it's under an IRB trial right now. And I was going to try to share my screen with you to show you the actual data, but I can read it off to you. You're familiar with the life length 20th percentile short telomere?
That's the one that we have historically used as well.
Right. So the reason this is important for the audience is because it's your short telomeres that are actually the most critical. Your average telomere length is interesting. Your median telomere length, or most commonly occurring telomere length, is interesting. And telomere lengths in the T cells here are a bell-shaped curve. There's some that are longer, some that are shorter, but it's the short ones that are actually driving cellular senescence. And so being able to improve your short telomeres, take your short telomeres and make them longer, is believed to have the healthiest benefits. And what we've seen, even with products like TA65 or TAM818 or Isogenis, is that they selectively seem to extend the short telomeres. They also have an impact on the median telomere length and average, but they seem to have a bigger impact on the short telomeres. So in February of 2020, I had my telomeres tested, and my short telomeres, 20th percentile short, was in the 65th percentile, okay, which, is reasonable. I had COVID, and post-COVID, it dropped down to the 29th percentile. Mm. And then after using the product for about 6 months, I now am off the chart. I'm actually in the 99th percentile. And if you were extrapolate over to where it comes back into the chart, I would be in about the 75th percentile for a 35-year-old, right? So pretty dramatic changes that occurred, right? I've tested since then, and I've had some degradation, but I went from a short telomere length of like 4.9 up to like 7.6, which is pretty dramatic, right? Kilobase. So anyway, the point is, for the audience, these technologies that can actually start to go after some of these hallmarks of aging, I think, are a key component to cracking the code on longevity. There's going to be more, it's a symphony. There's more pieces to be played here. We're never going to get one or two genes that actually, changes the whole thing. But
we wish.
But we wish. That's right. But we wish. And I think we'll become much more sophisticated in going about this as we go. So I'm really curious about the fact that you did all four. And have you done follow-up measurements with regards to Clotho with kidney function or blood vessel function, or some of the things we know that Clotho is particularly good at protecting?
I have really good kidney function, and I wish that I would have been tested for my kidney function before I took my dual gene therapy in 2015, because I actually think that telomerase reverse transcriptase was a game changer for me. I had chronic back pain whenever I wouldn't drink enough water, if I ran a certain distance, and I no longer had it after the first set of gene therapies. So I know there were benefits that unfortunately we didn't track with
Interesting.
before the first gene therapy.
So let me ask you this. So where did you have to go physically to actually have this done? Since it wasn't done in the US, you had to go someplace. Where did you go?
Right. So to do the first gene therapy, I went to Colombia because the co-founder of the company had a clinic in Colombia. And so we did it down there by consent. And then the second gene therapy that I did, I decided to try international waters, which I wasn't a huge fan of after the fact, but maybe under better conditions I might have been. And those were the two places that we went for my gene therapy specifically.
So just so I understand this, you helicoptered onto a boat, either that or you landed a jet on an aircraft carrier, or what?
No, we spent a very, very long time on a boat, traveling miles off the shore.
Okay, got it. Okay, so you jumped on a boat in a harbor and then went off into international waters. Okay. Got it.
And that was interesting. And it was something I wanted to try because I wanted to see what people might think of something like that. Again, with investigator-led studies, it's not necessary, but I just wanted to experience it, and I had an opportunity. So I said, yeah, hey, why not?
Let me ask you this. If you were going to do it again next week, where would you go?
I would go through integrative health systems. I would definitely go through one of their doctors, and they're different places of the world. So it would be who's available to treat me at that point.
Got it. Okay. And can you give us a ballpark on what the price is, what the cost is for people to do gene therapy like this? This is pretty cutting edge stuff. I've heard like $1 million in the past, but I suspect that's probably not the case. I don't know, or where is it?
Well, I think you hear that because the approved gene therapies in the United States start at $425,000 to treat one eye. And if your child is born with spinal muscular atrophy, you're going to pay $2 to $5 million. And those are really, really expensive gene therapies. Gene therapy itself is very expensive to make. It costs hundreds of thousands to make one gene therapy, but in medical tourism, probably to treat your entire body is less than treating one eye in the US.
Mm-hmm. So some hundreds of thousands of dollars is what you're saying.
Yeah, it's still going to be expensive, but it's not going to be as expensive as the drugs that you've seen in the regulatory system.
And is that the cocktail for all 4 genes, or is that?
I don't know really what I'm lucky to have participated in something that I don't know the exact cost of it, because I'm sure I've had a lot of people tell me, really be careful. They ask me not to drink and everything else because I'm a very expensive test subject.
There you go. That's right. Yeah. Take good care of yourself. Okay, cool. So your work here in the U.S. is focused on kind of working through the FDA to get FDA approval on some of these treatments here.
Yeah, but I also work in the regulatory side as well. So I have a company called Best Choice Medicine, and we work towards getting what's called pre-regulatory routes. They'd be regulatory routes, but outside of regulatory bodies, in order to get terminally ill patients access to this type of technology, because as you probably know, Right to Try does not include this class of drugs.
Okay, interesting. I was not aware of that because we function in Texas where we do have Right to Try,
yes,
which is helpful for us, quite honestly. But I didn't realize it did not extend to this. Okay.
Nope. It's only drugs that have been through a phase 1 trial. And most of the most promising technologies should have been through phase 1 trials a decade ago, but they are not because it costs an exorbitant amount of money to get there. And things are not incentivized for drugs that are game changers to an industry. There's a lot of worry about how it will really change how people work and, some of the large companies that employ a lot of people.
Sure. Yeah, it's really interesting how political, let's call it, all of this becomes political and economically driven. It all becomes so.
And yet the U.S. itself would save upwards of $30 trillion.
Right. Exactly. Yeah.
By slowing aging by 1 year.
Yeah. No, I mean, none of the math makes any sense unless you're actually collecting the dollars for disease management. Right.
Yeah.
So
it's just a known system. Companies understand how that works. We go to investors and, we have innovative ways to make money pre-regulatory route. And, people like to gamble their money on, vastly bad decisions. It costs $2.6 billion to make one drug, and there's a 94% failure rate in phase 3. And that's because we do an enormous amount of studies in animals that don't predict human outcome. So we are doing things wrong. And not us. We are saying, let's do things right. But historically, people are like, but that's the way we spend the money. That's how we do it, even if it's failing ideas.
Yeah, I heard Anthony Atala speak a few years ago. He actually ended up writing the foreword for my book, and he's basically the director of regenerative medicine at Wake Forest. And in his world, they basically have created, and they're not the only place, but where you have a body on a chip, so to speak. You can have a liver, you can have a heart, you can have a brain, you can have kidneys on a chip, so to speak, or you can put together a whole body, 4 or 5 organs or whatever. And what was fascinating to me was that there was a chemo drug, I forget the exact name, that had gone through all the phases of the FDA, was out clinically used for 10 years. And then it was discovered that it had a pulmonary side effect, right? People were dying from, I don't know if it was pulmonary fibrosis or some adverse pulmonary effect. So he took the drug and put it into his, I'll call it body on a chip. It was a lung on a chip, right? So think of this as a micro organ, if you're listening to this, like a micro lung, a human lung, but a micro lung. It comes actually from a biopsy from a lung from a human being. So this is real lung tissue. And they incubated the lung tissue with the drug for, I don't know, within 4 or 5 days, they figured out that it was pulmonary toxic, right? How many years of FDA approval, how many trials, how many studies, how many dollars, how many years in clinical practice, how many millions of dollars were spent, lives were lost, and they figured it out in a few days that this was toxic. And so we're actually working with him on some of the stuff that we're doing, right? So we're designing trials right now for this because we want to get to answers faster, so we can iterate
Well,
faster.
and that's the way forward. So Biden in last December signed into law that you didn't need animal studies for applying to the US FDA, but the US FDA still doesn't recognize other forms of testing.
Right.
And so, there's already been sign off that you don't need to do that, but there's no route forward not to do that. And there are about 18 companies that are doing like an organ on a chip type of technology that I think will get better over time and be more predictive. So yeah, it is exciting technology.
It is exciting. Yeah, and I know Atala's been working very closely with the FDA, and he's gotten lip service on some things, but of course that being translated into what's actually going to happen is interesting. Anyway, if you're listening to this, what's exciting is that we really do have ways to iterate, innovate, learn, and produce things at a much faster rate. And I think when you start to bring in AI and quantum computing and you start to put it through these biological systems that are very quick as well, I mean, I think we could really crack the code on all of this in 5 years or 10 years with, just getting away from these really kind of archaic methodologies, right?
And the best way to do it is human data. There's nothing that's better than that, actually.
That's right.
And, that's what we really need to push towards. And we need to push towards patient access and advocacy. I mean, for all the people who will be dying today, they could have been given the dignity to try a drug that might have saved their life.
Yeah.
And regenerative medicine will be best used, we can all admit, in healthier subjects, but it should be used in the most terminal to start with. And so, that's why I really push to best choice medicine, because it gives any country in the world a legitimate ethical and moral use of technology so that they can help people get advancements in drugs faster. I mean, one of the big evidences in that was that we had a COVID vaccine in 10 months of a new technology that hadn't been used historically in massive amounts in humans. And COVID was a terrible thing, but 41 million people will die this year of aging-associated diseases, and a bunch of children in which we know genes that could be beneficial to their condition.
Mm-hmm. Yeah, no, it's really an interesting landscape. It's super exciting scientifically, and it's can be frustrating and difficult when you try to apply it on this landscape, right? So that's always the tension, if you will. So what's next for you? What's where are you pushing into now? Is it more of getting the FDA approvals and trying to push forward for recognition on these things? Is that where most of your energy is going?
Well, we're pushing for 3 areas. I'm pushing towards the education of the advocacy of patients. So I'm pushing more towards people understanding and learning about medical tourism, because I'm seeing that these great unmet needs and these patients who need access need to go somewhere. And that includes pushing nonprofits to help facilitate payment for people who can't afford new and innovative therapies. We'll be pushing our dementia study towards the US FDA constantly. We just finished our pre-IND and got our response back from them. And then we'll push the CMV platform towards use. We may have a very exciting study to talk about later in about a month or so from now around that. And then, of course, I'm always working with a multitude of stakeholders to both educate and move technology forward so that we can advance the gene therapy space.
Awesome. Beautiful. Steve, any thoughts or comments as you hear all this?
No, I'm looking forward to getting to our Age Hackers Plus segment, because that first question, what do you think we're going to be doing differently 3 to 5 years from now? I'm really looking forward to that answer.
Yeah, that'll be a good one to get to. Well, Liz, we can't thank you enough for joining us. It's really been a pleasure chatting with you. I remember it was last time, and really fun to catch up with you and see all the things you're really involved with. Yeah.
Look what I have.
Oh, wow. Look at that. 100 is the new 30 right there. Beautiful.
Yeah.
up a copy.
Yeah, my husband used it and already ordered something out of it. So,
Okay.
I know that I got an opportunity to review the book and I was traveling and I got back and said, I'm going to get my review in, and I missed it. And I'm so mad about it.
Yeah. Yeah. It's been well received. It became a number one seller on Amazon fairly quickly. And then I get a lot of feedback. People say, oh my gosh, I didn't realize how comprehensive this book was, how good it was. So it's like, I don't know, not bad. So we're happy with that.
Not a bad job.
Yeah, thank you. Yeah.
Thank you for listening to this week's episode of the Gladden Longevity Podcast. If you would like more information on what we've discussed or other topics, please reference the show notes or go to gladdenlongevitypodcast.com. You can also find us on Instagram, Facebook, and Twitter by searching Gladden Longevity. If you've enjoyed this podcast, please subscribe to get future episodes delivered to you and share our podcast or this episode with someone in your life that you may find benefit. Thank you for listening. We'll be back next week with another exciting episode. The Gladden Longevity Podcast is provided for informational purposes only. It does not constitute medical advice. This content is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. The use of any information and materials linked to this podcast is at the listener's own risk.